Okay.
Okay, great. Well,
Uh-huh.
thank you so much for inviting me. As Laura said, I'm retired, but I'm still having fun trying to communicate science and talk a little bit about writing. And as Laura said before the recording started, I have published a book which maybe she can pass on to everybody, the ISBN number, and maybe you'd be interested. It's a little bit British centric or UK centric, but I think the messages are universal about where different species are from and the fact that once they're here, especially in the UK, they're part of our landscape and maybe we should accept them. I think Brazil has slightly different rules about where things can come from and what species can come out of Brazil, but it might be of interest to you. Right. I'll start on my talk now. The title is Finding Your Scientific Story by Writing Backwards. And just a little bit of background of this. It was originally an editorial, the paper that came from this, which I hope some of you have read, um, as me writing it as a language editor for Marine Life Sciences and Technology. Now, there's a little bit of a blurb here on the bottom right-hand side of this slide that says Marine Life Sciences and Technology provides a prime platform for new discoveries and theories in marine and aquatic, so they mean freshwater and estuarine waters, life sciences and technologies. I think I'm going to put a little bit of a plug here for the journal. It's a good journal. It's got a reasonably high impact factor of 5.7 roughly. I think that, well, I've got that in 2023, but it's around 5 or 6. Um, it's very open in its scope of information. If an organism is from an aquatic environment, they will consider it. It doesn't have to have a deal with specifically technologies or just ecology. It can be any number of fields. And I urge you to consider it. One of the other advantages of the journal, especially for people whose second language or third language is English, is that every paper goes through a language editor after it's been tentatively accepted. And that was my role, is to read over the paper and fix up the language or ask questions about the language or the structure. And so it's rigorously looked at for its English, which you still have to write a good paper and do the English reasonably well, but it's tidied up and polished by an editor. So that's an added step that they offer. So at least think about publishing there. Um, right, before I get, is that going to work? There, before I start on the talk proper, I wanted to give a couple of quotes. And this one is one of my favorite quotes from science, from Louis Pasteur, as in pasteurizing. Um, and he says, there's a translation from the French, or a rough translation from the French: chance favors only the prepared mind. Now, what does that mean? It means that if something happens in your studies, in your research, and you find something and it's surprising, it comes by chance, really only those people who have read widely and know lots about their subject and beyond their subject, they have a prepared mind, can see that chance as an opportunity. So this encourages us all to read much more widely across our field, but other fields also, and even the humanities if possibly, just to read very widely and understand things, go to lots of seminars, and all of a sudden you'll get a result in your science and your research, and you'll say, oh, that fits in with something I saw ages ago. And so you're prepared for it. And a good example of that, sort of a classic example of that, is Sir Alexander Fleming's discovery of penicillin, because at least the story goes that he was playing with bacterial cultures and some mold got into his cultures and it prevented the bacteria from growing. And here on the right-hand side, you can see a petri dish with some penicillin mold in it, inhibiting bacterial growth. Now, he wasn't planning to do that. This was a chance occurrence. And what he said, his quote, is, one sometimes finds what one is not looking for. So that's another way of saying it. But you only recognize the ability to find it if your eyes are open to opportunities. So back to that, chance favors only the prepared mind. This is a take-home message from this paper, and I'll pick this theme up later on in the paper or later on in the talk. Right. As I said, I hope some of you have read the paper and maybe we'll have some questions for me after I finish speaking about the paper that maybe I didn't even mention in the talk. One of the questions I often get asked is, who is this E.I. Montagnes, this Ian Montagnes? Is he your brother? Is he your son? In fact, he's my father. My father's now 93 years old. He was in his late 80s when we wrote this paper. Um, he's an editor. He was editor-in-chief of University of Toronto Press, but he's also been an international editor, working with non-English speakers across the world, and was very good about providing guidance on how to write simply and straightforward. And the other person who's written the paper with me is Zhao Yang, who's a Chinese collaborator I've worked with for years, an ecologist, and he has a good feeling for how to communicate in China with the Chinese. And much of our audience for marine life science and technology was China. It doesn't have to be, it could also be Brazil or any other countries, but he helped us make sure that we were getting across the message to the Chinese. So those are my co-authors on this paper. I'm going to start by sort of defining what a scientific story is. And story is an odd word. You sometimes think of a story as a fairy tale or a novel. I don't mean that. I mean, another word for that is narrative, a discussion or description. And usually a scientific story or narrative provides a problem and then solves that problem, provides a solution to the problem. So let me go on and tell you a little bit more about that. But I'm going to do that following the IMRAD formula. Now, IMRAD, I hope you all know, is introduction, methods, results, and discussion. The four main components of almost any scientific paper, what we're taught about as undergraduates and encouraged to follow all the way through our scientific careers. I'm going to pick up the IMRAD theme over and over again through this talk. So following the IMRAD format, we have a beginning. And our beginning has an introduction to it. And we could say maybe, what effect is climate change having on Brazilian wildlife? That's our introduction and the problem we're presenting in our introduction. Now, in the introduction, we might narrow that down and say, is temperature rise affecting forests in Brazil? And that's a problem. And we indicate why that's important for not just for Brazil, but the globe in general. And we produce possibly a series of hypotheses. We might even give a hint as to how we're going to test those hypotheses. So that's the introduction, the beginning of the story. The middle of the story, the center of it, has the methods, how we would collect data, how we would analyze those data, and then the results we get. And the results would be at least a beginning to the solution of the problem. Now, I'm probably not telling you anything you don't know, but it's important to set this all up so that you understand why I'm going to say it doesn't always work. And then finally, the end of the story has the discussion, and that would put the results, and maybe we found that temperature rises in Brazil are not going to be substantial to affect the wildlife, but in fact, the drought associated with those temperature rises or the rainfalls associated with it is what's going to affect the wildlife. And our discussion would put that into wider context, and it might even go one step further and say, what we now need to do is look at alternative sources of energy so that we reduce the amount of CO2 and solve the problem. Now, I've just made that up as I went along. That's maybe not a real paper, but it gives a feeling for the beginning, the middle, and the end following the IMRAD format. But critically, what I've done is provided a problem and ended up with some solutions. So the take-home message from this slide is a scientific story provides a solution to a problem. And if your paper or your thesis uh, or your talk for that matter doesn't do that, you're probably missing out on catching your audience's attention. And what I'm going to do here is show you a problem about writing and give you a solution to how to write. But before I do that, I'm now going to talk about putting writing papers into what I'm going to call the circle of science. Now, this circle of science is at least what funding agencies see as the theoretical way science works, and maybe your supervisor or university higher ups see the way science works as a circle. Now, let me explain what I mean by that circle. So the circle of science, it starts with us having some ideas and seeing some problems and writing a proposal. Just as I said about the Brazilian forests and climate change, that set out a problem. And it could ask some specific questions. Are mammals affected more than protozoa in the Brazilian forests? And I'd write a proposal about that, do my research carefully, my literature search carefully to find out what's known about the subject, find out what's not known about the subject, introduce those as problems that are questions that need to be addressed, and say exactly how I'm going to collect my data and how I'm going to analyze those data to answer the questions. If I'm lucky, I get funded. I think many of you are students, and so it wouldn't necessarily be you get funded, but you might write a proposal and your supervisor says, yes, that's a good question to ask, or those are good questions to ask. Those are ones that you can carry on with. So it could be your uh, uh, well, your research proposal for your student project. So funded or supported by your supervisor. The next step is you're all ready to go out and do your research, and you conduct your research, whether that's laboratory work or field work or in silico using a computer to model a system. You do that research. And you collect data, of course, and you analyze the data following the methods described in the proposal. Of course, your research is also conducted using the methods provided in the proposal. So it's all a linear progression. You've done your analysis. Your analysis provides you with your solution. That's the results. And you can develop some conclusions, which is your discussion. And you write the paper all about what you found. And that links the problem of it is produced in the proposal to the solutions collected by analyzing your data. You publish it, and then you can write another proposal on something similar to what you've been doing, or maybe an entirely different study, but you've got the credentials to write another proposal and get funded. And there's your circle of science. And you're incredibly happy. Woo! If we follow the IMRAD format, the IMRAD structure, our introduction of our paper is directly from our proposal, because we've written out why, we've defended why we need to do the work. We've provided the background for it. We've provided our questions. We're all set with introducing it, and that can go straight into the paper. The methods are straight from our proposal. How we were going to analyze the data, how we were going to collect the data, you can just copy that information and stick it into your paper. The results are presumably what you predicted in your hypotheses, and they're from your experiments that are based on your expectations, again from the proposal, so that should all flow nicely. And your discussion will be pretty much what you picked up in the introduction. You'll just expand on the introduction. You might, after a couple of years of research, have to go back to the field, the literature field, and read the literature and find out if anyone else has written more on your subject and add that into your discussion. But pretty much you can reiterate the questions that you're asking in the discussion. You can answer the questions, and then you can put them into a larger context to provide solutions. So it all fits together. Laura, I just saw you come up. Was there a question? Am I going too fast?
No, you're fine.
Okay.
No question.
Okay. Okay, great. Um, it all sounds perfect, doesn't it? It's all going to work perfectly. I can see Laura frowning a little bit. So the question really is, how often does that happen? This circle of science leading to a paper produced following the IMRAD structure. I've written a bit over 100 papers now, from anything from my own papers to student-led papers. Of those, what I just described to you has only happened one or maybe two times. It's been all worked perfectly that one or two times. And in fact, one of the times happened with a master's student. Everything worked perfectly. We published the work, and it ruined her for life because she thought that science worked perfectly. It doesn't work perfectly. From at least a sample size of one, me, the circle of science is broken roughly 98% of the time, maybe even more than 98% of the time. It just, it's a nice idea, but that doesn't work. Stuff happens. And what I want to talk to you today about is what we do when stuff happens. So there's our circle of science again. Um, what often happens, not always, it could be any number of steps. For instance, if you lived in the United States at the moment, maybe your funding got cut. And so the whole thing would fall apart. Sometimes your equipment breaks down and you can't do your research, or the weather's horrible and you can't go out and do your field work, so the research doesn't get done. But one of the very common problems is that you get funded, you do the research, you collect the data, but the data are just weird. They're strange data. And what I have here is a little demon or devil saying they're demonic data. They're not what you thought they were going to be. They're just a pain. Uh, and if you get these strange data, then you may not be able to analyze them the way you suggested you were going to do in the proposal. So your analysis falls apart completely because the data just don't match up with the analysis. If the analysis doesn't work, then the problems you provided in your proposal don't necessarily get answered. So your problems disappear, and the problem solution component of your study has gone, and the entire paper falls apart. So to summarize that, the questions in our proposal can't be answered by the analysis of our results, and the whole paper falls apart. And as you can tell, you pull out your hair because I've done that completely already. Those are those other 98 papers that I've written and it didn't work out right. And if that's the case, we can't use the IMRAD structure to write forward. I'm just going to have a sip of tea here for a second. So what do we do? Well, as I said, I'm here to provide a solution to this problem. And that's by doing something that I think most scientists do, or most biologists definitely do, all the time. We don't always admit it, and we certainly don't admit it to undergraduates. Um, but it's worth telling you as graduate students, postgraduate students, how you can deal with this. And a few times when I've given this talk, I've had people in the background just saying, I can do this. That seems like cheating. And we can talk about that a little bit more, but it's not cheating. So how do you fix the circle of science? The first step is a little bit of a change in mindset. And that's to look at the strange data, but look at it as an opportunity, as chance favors the prepared mind. And instead of it being demonic and evil, look at it as happy pink data with maybe a fairy godmother looking over it. This is an opportunity for you, these strange and unusual data. And think about the data. And then after you've thought about the data, you might be able to do some new and very different analyses And you'll only be able to do those different analyses if you have a wide understanding of the data and how it could be analyzed by other people, not just your data, but any number of sets of data. And this is where really preparing yourself is important. If you analyze the data, you might get some surprising results you never even thought you were going to get. And you need to think about those results, and they may provide new solutions, not the solutions you suggested in your proposal, but completely different solutions. So you have to think widely about that and say, oh, that could be useful for somebody. And here's the trick. And this is what surprises people sometimes. You've got your solutions already. Now you have to phrase those as problems that need to be answered. So this is where writing backwards comes in. You start off with the answers and you then provide the questions or the problems. And lots of us do this. You then write a paper with your new problems and your new solutions. You publish it, and then you can write another proposal, which doesn't work again, and you have to do this again. Or maybe it does work, and maybe you learn from this to write better proposals. And we're all happy. So what I want to do next is talk a little bit about these areas in yellow. But before I do that, Laura, are there any questions, or should we stop for just a second to see if anyone has any questions?
In the chat, you don't have anything.
Does anyone want to ask anything
Yeah,
at this point?
yeah, if somebody wants to ask a question, can ask now before we go on. No?
Sure. No, I'm just curious about this because I don't have much experience with this process. So doesn't that I guess you'll talk about it, but doesn't that break your sort of get funded side of things? How does that work?
I don't know what it's like in Brazil, but most funding agencies, uh, at least in the pure sciences, maybe not so much in the applied sciences, but in ecology especially, they don't really care if you don't answer what you've asked in the proposal. They want you to get outputs that have impact. So
Got it.
if I write some papers and those get highly cited because they provide some really exciting stuff, funding agencies are thrilled. You succeeded in doing that. And most funding agencies, at least at their roots, have scientists who know this happens all the time anyway.
Hmm.
So
Got it. Because my experience is more from entrepreneurship, and I found myself trying to fulfill goals that make no sense for the business anymore. But you just gotta fill the goals that you've been set
yeah.
by the funding agency. Yeah.
Yeah, okay, yes, I think probably more applied that this would be a problem unless I have no experience with that, so I don't know. But if you could convince them that you've found, for instance, if you found penicillin, I don't think they'd complain. Uh,
Yeah, sounds right.
but yeah, but good point, really good point. So I guess I should limit this to more of the pure sciences in academia than money making. But
Thanks.
that's great. Any others?
Yeah. Any other question? No. I think you can go ahead.
Okay.
Okay, thanks.
So now we've given up with writing forward. Let's look at writing backwards. So in writing backwards, the way I argue in the paper, here are the steps. And the first is to look at your strange new data, the data you did collect, and try and find some trends in it, find some conclusions. Now, I think I've said this, yeah, this is the most challenging step. And the way I do this, and I've suggested other people do this, is do any number of analyses. Analysis of variance, principal component analysis, maybe only use a subset of your data that actually has some kind of trends in it that show something. You might end up having to throw out half of your data because it just all went funny, but there's some other trends you weren't expecting in it. Or you might decide, if I just do a couple more little experiments, I can build on this and really develop this new theme. So what I've got there is a whole set of pictures of different analyses that could be done. And this point takes a fair bit of creative thinking. But eventually you'll come look at it, hopefully, and find some conclusions. Let me give you an example. We were going to use, like Laura, I'm a protozoologist or protistologist, and back in the 30s, there's a guy named Gauss, who's a mathematical biologist who did population cycles, and he looked at one protozoa, Paramecium, eating yeast, and looked at population cycles as a result of the protozoa growing up and then the yeast dying off because it's eaten, and then the protozoa die off and the yeast grow up, and then the protozoa grow up, and there's population cycles. I thought that's a great system to use. And we did some experiments to see if we could get the protozoa, the Paramecium, to eat the yeast. And it ate the yeast because it's not very smart. It gobbled it up, but it didn't live on it. They just died. And we tried tons of different yeasts on this. So we kept on trying. Didn't work. They just didn't grow on it. So we scratched our heads and tried to figure out what was going on, and decided that probably the original studies by Gauss back in the 30s, he didn't have antibiotics and he had bacterial contamination in his cultures. And it was actually the protozoa eating the bacteria and living, and eating the yeast as just you might eat golf balls or something like that, or some plastic stuff that doesn't get digested, but it's the bacteria that we're allowing it to grow. You might think at that point we'd just throw out all the data and say, well, that was an interesting direction. I give up. I'm not going to publish that. But we thought, no, people should know this. So we wrote up the paper as a conclusion that we tried all this work to get the yeast to be food. It wasn't food, and recommended that people never follow this work, and suggested that Gauss had an infection in his cultures. Now, of course, we titled that paper, "Did Gauss Have a Yeast Infection?" And I now get all kinds of medical interest in my work on yeast infections, which isn't true at all. But still, we published the work because we turned it on its head and got a publishable unit out of it. So the work wasn't wasted. Now that's a very simple example, but and I'm sure you can think of other examples that or other problems that you have that could be solved much better. But this gets to the point of Louis Pasteur saying chance favors only the prepared mind. You look at all the data, all the analyses you can think of, you talk to people about it, and you may find some way of salvaging your work, and not just salvaging it, making it even more interesting than you thought it was going to be. So my suggestions here to do this, and I've done this with many students over the years, and I bet lots of people do this, is to look at all the data in all the ways you can possibly think of. And one recommendation is make good figures. Now, here I have several different ways in which figures could be made. They could be just handwritten, but so that anyone can understand them. They could be done beautifully using a graphics package so that everybody could understand them. Or they could be done on computer screen. Again, nice graphics. But you make lots and lots of figures and tables, and you can then examine them, spread them all out, and I've had students spread them all out all over the floor and try and look at them to figure out what story we're going to tell. And sometimes, well, let's see what I've got here. Spread out the figures, get together with a group or your supervisor and your friends, and show them all the pictures, because somebody might have an idea of what could be done that you hadn't even thought of. You don't just have to have all the ideas yourself. You can use other people's ideas. Call a friend. And then this is critical. You don't have to use all the figures you've made. In fact, you probably shouldn't. A lot of the times, you're going to throw away half or 3/4 of the figures you've made if you've done lots of them and are having all kinds of ideas, and keep the ones that have important conclusions that tell a story. So put the other data aside, save it for another day, put it in an appendix of the paper or supplemental material if you really want to, or just hold on to it and think about it. And maybe it'll spur on some new ideas later on. And critically, well, and then use those figures, the ones you've kept, to tell a story. And it might be that the work you did at the beginning doesn't fit perfectly as the beginning of your story. It might come to the conclusion of your story. So you can take these figures and move them around. As scientists, as biologists, we're often very visual people, and recognizing the trends that are shown in our figures and our tables is the best way to put your story together. At least for me, that is the case. And finally, just a word of caution, maintain integrity as you do this. Now, what do I mean by that? Let's say you were doing your study on the effects of temperature on biodiversity in Brazil, and you looked at 5 study sites, and 3 of them told one story, and 2 of them told another story. It would be a lack of integrity to only tell the story with the 3 because it's a sexier story. It's a more interesting story. If the other 2 sites tell some other story, you need to figure out why, and you need to include that in your story possibly. And maybe contrasting the 2 points is more an even more interesting story. But don't just ignore possible trends in important information because it doesn't tell a good story. That's a fairy tale, and that's inappropriate. And that's not maintaining integrity. So that's what I mean by that last point. Okay, so now you've got all those figures. You've reduced them down to just maybe 2 or 3 figures that really tell your story. And then this was where writing backwards comes in. You've got your solutions, you've got your conclusions, and in fact, you've probably got your key questions that you're asking too. From those 2 or 3 figures you're going to use, or maybe and tables, write the results up carefully associated with them, and just the results associated with those figures. You may have spent 4 years doing a whole bunch of work, and there's lots of other stuff you've written up results as you went along, but maybe you just want to put those aside and only stick with the results associated with the figures and the data you're going to present. And people often forget that. And I often, as an editor, read over a paper and read some results and say, why are you telling me this? This has nothing to do with your conclusions or your key questions. Get rid of those other results. Put them aside. Likewise, the methods you write are the methods only associated with the results you present. Don't tell the reader anything they don't need to know. Only tell them about how you did the methods to obtain the results that you're presenting in the paper. Again, we all want to tell everybody how hard we've worked, but the reader doesn't care about that. That's your ego wanting to say, I worked really hard and did all this stuff. Stick with just the methods associated with the results. And then comes the next most difficult part, and this is the creative part. I tend to write my discussion and my introduction at roughly the same time. I maybe start with my discussion and provide my take-home messages, my conclusions of what I've found. But as I'm doing that, if I'm using Word, Microsoft Word, I split the screen. And I have a heading at the top of the screen and the top window saying introduction and the bottom window saying discussion. And if I write a conclusion in the discussion, I then go up to the introduction and write the question that relates to that conclusion. And I continue doing that by writing the 2 together. And as I flesh out the discussion, I think, oh, there's some terms I need to introduce in my introduction because I'm going to talk about them in my discussion. So I then make sure I make notes to myself, or I really write out those terms, I define those terms or concepts. And so writing the introduction and the discussion is dealing with all the bits that came above it, but it's done iteratively, where you add bits to one and then add more to the other. And sometimes there's going to be places where you decide that a detailed explanation of what you're doing fits best in the introduction rather than the discussion. Or you may decide that you've talked way too much about something in the introduction and you're going to bore your reader, so you take some of that and you move it down to the discussion. And that requires a lot of thinking and crafting the paper so that it reads well. And often your supervisor and other readers will help you with that. And I often get many of my colleagues to read over stuff and say, no, David, you haven't explained that adequately in the introduction. Or I'm getting really bored by reading your introduction and you need to cut it down and remove some of that stuff and put it in the discussion. So get other people to read it. So what I've hopefully done there, with lots of little bits of sidelines and information, is shown you how when you get strange data, you can analyze it to get surprising results, interesting results, get new solutions. You have new questions and you can write a paper and publish it. In other words, classically and as undergraduates, we're taught that a scientific paper has a beginning, which is the introduction, a middle, the methods and results, and an end, the discussion. And we've written that as a problem to solution, and we followed the IMRAD structure, and that's the story. But in reality, most of us, or many of us, start off with the solution because it's a solution that we weren't expecting, and we write backwards and present the problems and how we solve those problems. So you often, when you're reading a scientific paper, and I'm sure this is true 75 or 85% of the time, especially in ecology, when you're reading a scientific paper that just seems like it all worked out perfectly and they have a great story and it all followed the IMRAD structure. In fact, they wrote backwards just the way I've described. So I'll stop there and answer any questions or address any issues. Thanks very much for your attention.
Thank you, David. It was excellent as always. So now we are open for questions.